Now, in our last meeting, we started looking at some fundamentals of enzymology. So we first looked at what enzymes were. We said enzymes were mostly proteins, but of course, you could have some catalytic RNAs, which we refer to as ribozymes, which take part in the processing of RNA molecules themselves in their restriction, some form of recycling of these molecules. And we talked of enzymes as being biological catalysts that speed up biochemical reactions in biological systems. And this is essential for the sustenance of life because without them, the biochemical reactions in biological system wouldn't be as fast as they should be so that they will not be able to have uh, that coordination between them so that there will be that sustenance of life. So they are really essential for the sustenance of life. We went ahead and looked at the activation energy profile of chemical reactions in general and compared it to enzymatic reactions. And we looked at the blue line showing a reaction profile for uh, a non-catalytic reaction. So it's a chemical reaction which is occurring without a catalyst. And we compare that to that which is the orange line, which is representing the enzymatic reaction. And we talked of the fact that all chemical reactions occur, with the same principle that reactants at a certain energy level will have to be activated to a point where we refer to as a transition state before they react to form a product. And that energy which is required to activate the reactants, the molecules, in order for them to reach a transition state, that energy we refer to as activation energy. And we said what enzymes do is that they reduce the activation energy which is required for the reactants to attain a transition state for the reaction to occur. And by that, the reaction is able to occur within a short time because the amount of energy which is required is less. And so the reaction occurs at a faster time. Then we went ahead and looked at some common terminologies in the study of enzymes. We looked at terms such as cofactors, coenzymes, apoenzyme, the active sites of enzymes, the process of activation, and even activators, which are the substances that will cause an enzyme to start to catalyze a reaction or speed up the rate, speed up the activity of the enzyme. And the opposite of that was inhibition, and the substances that will reduce the activity of an enzyme or completely even block the enzyme from acting. Those substances we refer to them as inhibitors. And then the substances on which the enzymes will act, those are the reactants. We refer to them as substrates. We went ahead and looked at uh, the system of naming of enzymes. And we looked at the naming based on the substrate on which the enzyme is acting, so that if the substrate is urea, you only add a suffix ase, ACE, to the name of urea to be urease as the name of the enzyme. And also, if it is lactose, we had that example as lactase to be the name of the enzyme. We also looked at the fact that enzymes can also be named after the reactions that they catalyze. so that an example such as DNA polymerase will catalyze a polymerization of nucleotides. And that is where the name is coming from, because the reaction is a polymerization reaction. That is, monomers, which are the nucleotides, are brought together to form a nucleic acid strand, which is a polymer. And that polymer is DNA. And so the name of the enzyme was DNA polymerase. The process of removal of a carboxyl group from pyruvate, a common reaction which you see in the metabolism of carbohydrates, is also catalyzed by the enzyme pyruvate decarboxylase. So the reaction was decarboxylation reaction. So the name comes after the type of reaction that the enzyme is catalyzing. Similarly, an enzyme that is acting in the process of the lysis of bacterial walls is also called a lysozyme, after that reaction, that lytic reaction, which causes the breakdown of bacterial walls. Then we saw some common names as well, and a common name as pepsin, which simply is from a Greek word, pepsis, which means digestion, because it was one of the first enzymes that was discovered, and it was discovered in the digestive fluids. So it was known to be acting or participating in the process of digestion. And so the name pepsin was given to it. Then trypsin, which was named after the method by which it was obtained for the first time, which is to extract it in glycerin. And that extraction into glycerin was done just by rubbing glycerin on pancreatic tissue, so that the enzyme just got extracted into glycerin by that process of wearing off of the tissue of pancreatic tissue. So that is after the process by which it was obtained. So these do not follow any systematic way of naming. Then we went ahead and looked at classification of enzymes. We saw 6 classes of enzymes. The first one was the oxidoreductase, which is involved in the catalysis of oxidation and reduction reactions which you commonly refer to as redox reactions. We went ahead and looked at transferases, which are enzymes that are responsible for the transfer of functional groups. So one functional group is transferred from one molecule to the other. We looked at this example of aspartic acid and alpha ketoglutarate, and we saw that the amine group from this carbon number 2 gets transferred onto alpha ketoglutarate, also on carbon number 2, whilst you have the ketone group, which is at the carbon number 2 on alpha ketoglutarate, now gets transferred onto the residue of aspartic acid to now give you products oxaloacetate and glutamic acid. And this reaction, I said, we will see it in the metabolism of proteins. And clearly you will see that this is a connection between protein metabolism and carbohydrate metabolism, because you see clearly that, yes, alpha ketoglutarate you will find in the Krebs cycle. Oxaloacetate, yes, too, you will find it there in the Krebs cycle. So you see that there is this process of transamination, which we will see in protein metabolism, and you see this specific reaction over there. And the enzyme which is involved there is a transferase. Right. Then we looked at the hydrolysis. It said they catalyze, as the name suggests to you, they catalyze hydrolysis reactions. I mean, reactions that are the breakdown of a molecule into two or more uh, new molecules by a hydrolytic reaction. That is by the action of water in that reaction. And we went ahead and looked at lyases, and these are uh, enzymes that are involved in either the removal or addition of groups in onto molecules to either uh, remove a double bond or form double bond, so that if the reaction that we are looking at was in the reverse reaction, then it will be that there was a removal of uh, water molecule from isocitrate to now give you cis aconitate, then the double bond is formed. But in the forward reaction, we see that it is rather the addition of a hydroxyl and a hydride from the water molecule that removes the double bond from the molecule to now give you isocitrate. And this reaction too, I mentioned to you, is one of those uh, intermediary reactions that we'll see also in the TCA cycle or the Krebs cycle. We'll see it there. And the enzyme involved there was aconitis. Then we saw isomerases. And isomerases, as the name suggests, they catalyze isomerization reactions. That is the change in structure of molecules that are isomers of each other. So they will have the same molecular mass, but just that they have different structures. And that process is an isomer. It's a process of isomerization, and so you have the enzymes classified as that isomerases. And then the last one was ligases, one common process in which you have seen ligases was in DNA, either you in the process of replication, that you have DNA molecules being reproduced, and after that, the nucleotides that come together to form a new nucleic acid strand are then joined together. So these ligases are the enzymes that catalyze the joining of two molecules. So we saw these classes. Then we went ahead and looked at the classification of enzymes based on their specificity. So what we just finished looking at was that classification was based on the type of reactions that the enzymes catalyze. But now we're looking at the specificity of the enzymes, on what substrate specifically does the enzyme act on. And by that classification, we saw absolutely specific enzymes, and these absolutely specific enzymes, they act on only one, just one and only one of a substrate, so that the name of the enzyme is after that only substrate that they act on. So the example which you looked at earlier, which was lactase, clearly tells you that the enzyme acts only on lactose. If it was acting on any other substrate, then it couldn't have been called lactase, because then the name will be misleading. So also glucokinase will be acting on the substrate glucose and only glucose. And I mentioned anytime you see the name of an enzyme with the word kinase in it, it only means that that enzyme is involved in the transfer of phosphate group from one molecule to the other. In this case, it will be involved in the transfer of phosphate group from ATP onto glucose to give you glucose phosphate, maybe glucose 6 phosphate. Right. Then we looked at group specific enzymes, which means that the enzyme will not be acting only on a single substrate, but it will act on a group of molecules. So if you take enzymes that act on, say, hexoses, hexoses are 6 carbon sugars. So that's a group of monosaccharides that these enzymes will be acting on. And the name is a hexokinase. Again, the kinase means that the enzyme catalyzes the transfer of phosphate group onto hexoses. So it can be any hexose, not only glucose, but it can also be, let's say, mannose, tagatose, cyclose. So many of these hexoses, you have these enzymes that are acting on them in the transfer of phosphate groups onto them. Then we also saw, say, pepsin. So pepsin, for instance, it will be involved in the breakdown of peptide bonds that are formed between the carboxyl group of amino acids with aromatic side chains, and then another amino acid. So a peptide bond that is formed between an aromatic amino acid and another amino acid. It could be another aromatic, or it could be a different amino acid, not necessarily aromatic, but there must be an aromatic amino acid in that peptide bond formation. Then you will have pepsin acting on that peptide bond. So that is specific because we don't have only one amino acid with aromatic side chain. We have tyrosine, we have tryptophan. So I mean, you can have these. We have 3 of them. We have phenylalanine, we have tyrosine, we have tryptophan. And so these 3 of them, when they form the peptide bond with another amino acid, then that peptide bond will be pepsin can be acting on that peptide bond, on breaking it. Trypsin, on the other hand, will be acting where you have again lysine, arginine. These 2 amino acids have amine groups in their side chains, and so that is what distinguishes them. So that yes, when there is a peptide bond formed between their carboxylic acid group with the amine group of another amino acid, that bond is specific for the trypsin will be acting specifically on those bonds to break those peptide bonds in proteins. Right. Then the very last one which we looked at was bond-specific enzymes. So that is a little different from what we just looked at. What we just looked at was talking about group specificity. So it's a group of molecules on which these enzymes can be acting, but this is talking specifically only about bonds. So for instance, you have say our salivary amylase that can hydrolyze alpha-1,4 glycosidic bonds in starch, break it down all the way to first of all you have maltose, and then later you can break that one down into glucose, so that you have that as energy source of the body. But it cannot do same for say cellulose, which is also a polysaccharide, but this polysaccharide is having beta-1,4 glycosidic bonds in it, and our salivary amylase cannot break down those beta-1,4 glycosidic bonds. And we saw the example of termites that you see termites are eating wood. It isn't that the termite is having an enzyme to break down the beta 1,4 glycosidic bond, but it's just that the termite is having a symbiotic relationship uh, with a bacteria called trichonympha, which is in the gut of the termite. So it is that bacteria actually that breaks down uh, the cellulose, because it has that enzyme cellulase that can break down the beta 1,4 glycosidic bonds. So there is also that classification of enzymes based on bond specificity. Then lastly we looked at the stereochemically specific enzymes. We talk of the fact that an enzyme may be acting on the L isomer of an amino acid, but not on the D isomer. And I mentioned that actually, naturally, most amino acids exist as L in their L isomer forms. And also the fact that as we go ahead and when we come to look at the metabolism of proteins, you will see that even where you have, in some rare cases where there are D amino acid uh, present, you see that there will be a convection of that D amino acids into L amino acids before they get you know uh, metabolized. They go through that process of transamination and deamination. Well, so in some cases, there are some enzymes like the D-amino acid oxidase that can, uh, you know, participate in the metabolism of such amino acids. Okay, so that was then. I think we ended with this. We ended with this practical application of our knowledge in enzyme action, and we look at the example of succinylcholine, which is used as a muscle relaxant because it's a bigger molecule that when it attaches to the muscle receptors, it takes time for acetylcholinesterase to hydrolyze it, compared to acetylcholine, which is the the original neurotransmitter, which is involved in the contraction and relaxation of the muscle. But when you have a bigger molecule like succinylcholine that gets, you know, attached to the receptors of the muscle cells, it takes a longer time, for it to be hydrolyzed, and thus you have the muscle being relaxed for minor surgeries that can be conducted, and by the time that the surgery is finished and the succinylcholine is hydrolyzed before acetylcholine takes over uh, to 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 be uh, attached to the receptor for that contraction and relaxation process to resume. The surgery would have been done so that you prevent a lot of blood spillage in the process of surgery. So we saw that as a practical example of our knowledge in enzymes. All right, now uh, we'll continue with coenzymes and cofactors. Just some fundamental knowledge of it, which is, as you see on your screen, the term which we looked at, apoenzyme. So this structure uh, represents an apoenzyme, that is the protein portion of an enzyme. And the last time I mentioned that the coenzymes or cofactors are required by the enzyme for them to become active. So when they are not attached to the enzyme is not active. So you see it being depicted here, the enzyme, that is the protein part of the enzyme, and then you have a coenzyme or cofactor. You remember I mentioned the last time that in some chemistry books you have coenzymes and cofactors used interchangeably, but in some they are separated. Some they refer to the enzymes as biological molecules, but they are not proteins. That's when they get attached to the enzyme, they cause the enzyme to be activated. And the cofactors are inorganic metal ions like copper, magnesium, calcium, and all of that. So we'll take a look at it further. So you see here that the enzyme, the protein part, you when the cofactor or the coenzyme gets attached onto the enzyme, it becomes a hollow enzyme. Call it so. That is a whole enzyme or an active enzyme. Here it is inactive. Where the cofactor has not attached to it, it is inactive. It's just the enzyme. But when you have the coenzyme now attached to it, coming to get attached, when it gets attached to it, you now have it as an active enzyme. And that active enzyme is referred to as a hollow enzyme. Now, as we said earlier, you have the cofactors being some inorganic metals. So you can have, for instance, copper ions that are required by an enzyme such as cytochrome oxidase. This is as the name when you see the name, you can tell clearly that this enzyme is involved in an oxidation reaction. You also see that, okay, of course, even the name, the cytochrome, when you see the name cytochrome, it only means that this is an enzyme that is having a structure of a protein, and then there is some prosthetic part of it, which is the heme, just like hemoglobin, except that hemoglobin is made up of 4 subunits of proteins heme uh, with heme being the prosthetic part of it that is having iron in it. Cytochromes can have different metals. So in this case, what it means is that the metal that will be there is going to be copper. So when copper is there, then the enzyme is going to be active. Similarly, you have iron, both the ferrous and ferric ions, the Fe2 plus, Fe3 plus, that act also as cofactors, again to cytochrome oxidase, to other enzymes such as catalase peroxidase, pyruvate kinase. You will have the metal potassium ion. Magnesium ions will be acting as cofactors for hexokinase. Glucose 6-phosphatase, pyruvate kinase. So on and on, you see these examples of metals: manganese ion, molybdenum ion, nickel ion, selenium ion, zinc ion. All of these are acting as cofactors to different to different enzymes. Now, when you come to the coenzymes, so you remember I told you cofactors generally will be referring to the metal ions, and the coenzymes are referring to biological molecules, but they are not proteins. But when they get attached to the enzyme, they cause the enzyme to be active. Now you have coenzymes being biocytin, coenzyme A. I think one of the commonest you have seen a lot of times in biochemistry was coenzyme A, which you saw in different biochemical reactions in metabolism. Krebs cycle, in even in glycolysis, and all the uh, metabolic reactions that occur, the metabolic pathways along the line, you will find coenzyme acting, especially if you take a Krebs cycle, for instance, the very first reaction, you see coenzyme coming in, before there will be that formation. So from oxaloacetate up to isocitrate, you will see coenzyme acting in that reaction. Now you see that at the right hand side, the right the at the end of the table, right hand side, where you see dietary precursor mammals, that you can relate to much more than the left hand side. But all of these coenzymes actually will be looking at, so if not all of them, some of them much more into detail, where we'll look at the structure of the coenzyme, and we also look at the type of reactions in which they are involved and how they actually carry out these reactions. That is their mechanism of action of these reactions. We'll be looking at those things. So, for instance, we will look at the structure of, say, um, coenzyme A, how coenzyme A takes part in a chemical reaction, which part of it is active, that actually will react with a substrate. We will look at biotin, which is the precursor to this coenzyme biocytin. We'll look at the structure of it and how it carries carboxyl groups from one molecule to the other. We'll see that. We'll see, for instance, I think folate as a tetrahydrofolate. You commonly hear of folate as folic acid, which is sold, in the pharmacies as a supplement, required for especially the development of neural tubes in in fetus. So when you have the fetus developing in the womb, you require adequate amount of folic acid, in that process. So you see that a lot of times folic acid is being prescribed for expectant mothers, because it is required for the development of the child, because it is one essential uh, one essential coenzyme, or call it a compound, which is required in the process of proliferation of cells in the body. We also see pyridoxine in the form of pyridoxal phosphate, and you see that also in the metabolism pathway of proteins. So we'll see pyridoxal phosphate involved in the process of transamination. That is the transfer of amino groups from one molecule to the other. We saw that reaction earlier with the transferase, like the transaminase or transferase. Now and then you also see thymine, which we see it as thymine pyrophosphate, which is also involved in the transfer of aldehyde groups in biochemical reactions. Then you will notice that a lot of these coenzymes are vitamin Bs. So you are seeing that the precursor here is vitamin B12. You see another one here, which is vitamin B2, and you see another one which is B6. You see another one which is B1. And you even see nicotinic acid, which is niacin. So these are vitamins. Then you begin to relate to it that, so all of these uh, vitamins are actually required for so many chemical reactions in the body. And these chemical reactions are actually tailored towards metabolism. So it can be metabolism of, say, drugs in the body. So you see that a lot of times when uh, you know drugs are being prescribed, when medications are being prescribed in the hospitals, you see that you have these vitamin B complexes that are prescribed in addition to these medications, and that is because the body at the time when in illness, is low in some of these vitamins, and whatever medication which is being given to you, the body needed some boost in order for it to be able to metabolize these drugs. And so that is why you see that a lot of times you have these B complex vitamins being prescribed in addition to some medications. Right. So we continue and look at some factors that affect enzyme activity. So the first we will look at is um, the concentration of the enzyme itself in, uh, say, a reaction system, or if you let's say even if you call it a reaction mixture, the amount of enzyme which is in that mixture, or the amount of enzyme which is present in that uh, reaction system will have the effect of proportionality, such that the more the enzyme, the higher the rate of reaction of the enzyme. And so you see that the graph plotted, or which is the rate against the enzyme concentration, shows that straight line, showing that sort of proportionality, direct proportionality between the rate of reaction of the rate of reaction to the enzyme concentration. So what simply it means is that if you have some amount of substrate in a solution and you have that substrate undergoing a certain chemical reaction, when you add an enzymatic, when you add an enzyme solution to that uh, solution, it will increase the rate of the reaction. And the more enzyme you add in there, the more the rate of the reaction. So if it was a reaction that is giving you a color change, it means that the more enzyme that you add in there, the more the color change will occur faster. So you begin to have maybe a deeper color when you have more enzyme in there, because you have more of the substrate reacting at a faster rate, so that you now will be able to see this difference in color occurring with the solution at a faster rate. So this is used actually in the labs that we run some enzymatic reactions, and you can actually look at, say, different concentrations of the enzyme, and then you measure the amount of light which is passing through the solution at different times, just using a spectrophotometer. And then you'll be able to tell that, okay, if you add this amount of enzyme, this will be the color. Like the color, when the color is deeper, you have less light passing through. When the color is, that is if you are measuring transmittance. But if you are measuring absorbance, then when the color is deeper, absorption will be higher, and you'll be able to have this relation as you see here, the graph that you see as the enzyme concentration against the rate of the reaction. You see such a straight line. Then we have also the substrate concentration. So what we looked at earlier was the enzyme concentration. But when you come to substrate concentration, you see that that relationship plateaus along the line. It tends to level up. And why is that? Is because you have enzymes. Remember, we talked about, say, active site of enzymes where reactions are occurring. Or even if you don't say specifically about the active site, just look at the enzyme as molecules that, when they are present, they are forming complexes with the substrate molecules to form a product. So what it means is that if you are adding more substrate molecules into a reaction system, and it comes to a point where the substrate is now more than the amount of enzyme that you have in there, it means no matter how much of substrate you are pouring in there, you don't have enzyme molecules available to react with those substrate molecules. And so you see that there will be that saturation of the system. There are enzymes in there, all right, but the enzymes are occupied by molecules of substrate already. So that even if you are adding more substrate, the enzyme molecules are engaged, and so they will not be able to react with those extra molecules of substrate which you are introducing into the system. So that is why you'll be having such a saturation curve. You see that the curve levels up, it plateaus in that manner. That we refer to as a saturation curve, or to show substrate concentration against the rate of reaction. Now, another factor is temperature. Temperature. Now, generally, you know that our body temperature is 37 degrees. And why is that? Now, you also realize that some of the reactions which you run in the lab, I think when you were doing some DNA extraction stuff, like you did some heating of the system, and you heat up to some 30 degrees, 35 degrees thereabout, and then you stop. Okay, when you are using, say, the how do you call it, the TAC, that is the thermophilic aquatic enzyme, which you use along the line for, I think, one of the reactions that you were running in the lab, I think even in your HND, was the polymerase chain reaction. You were taught of using the TAC, which is thermophilic, which you can heat the solution up to about, say, 70 degrees. And that is because that enzyme can actually, act at that level. But the enzymes that are in our bodies, really, they will be acting optimally at a temperature of, say, 37 degrees Celsius. Now, remember we said that most enzymes are proteins. So if most enzymes are proteins, it means that if you increase temperature beyond optimal temperature, those proteins are going to get denatured. So you see that this curve is showing that somewhere along the line, almost just like, say, 37 degrees Celsius, you have optimal activity or optimal rate of reaction. The maximum rate of reaction is occurring at that temperature. But when the temperature increases to 40, 50, you see the rate of the reaction is reducing. Yes, the rate of the reaction is reducing because of the explanation I just gave you, because the proteins, that which are the enzymes, will begin to get denatured. And when they are denatured, they become inactive. They will no longer be able to catalyze those reactions. That is why having your body temperature getting elevated at like body temperature going beyond 40 degrees is not a thing to joke with, because what it means is that the proteins in your body will begin to get denatured, and then you begin to have various things happening, like, say, your red blood cells will begin to break down. So, and when that happens, you can relate to the fact that then it means that there's going to be lack of oxygen that is going to the tissues, and many other proteins that will be getting denatured will be affecting many other biochemical reactions that are required for metabolism in the cell. And that, in fact, can result into fatality when the temperature goes really, really high. Now, how about when the temperature is below the 37 degrees, which is the optimal temperature. Now, when temperature goes below, it slows down the reaction. It inhibits the activity of the enzyme. And in fact, this knowledge is what is applied in, say, preservation of foods. So, what causes spoilage in foods? A lot of times it is fungal or bacteria, depending on the amount of water which is present in the food. So, because fungi will grow within a certain limit of moisture. Say, if you have maybe about 20 to about 35% of moisture in food, that medium is going to be conducive for fungal growth. And how will fungi cause spoilage of food? To cause spoilage of food, because it feeds on the food and then it releases its byproducts into the food, so that it is the byproduct which is a toxic which is toxic, or which is a toxin to you who goes to consume that food. And you can attest to the fact that, for instance, if bread starts to get spoiled, when you take a piece of it and you taste it, you see that there's some kind of bitterness in it, because the fungi had started to release toxins into that bread. So it is actually the byproduct of metabolism of these microorganisms that cause the spoilage of the food. Now, if it is their metabolism which is causing the spoilage of the food, then it means that you can lower the activity of the enzymes that are in those microorganisms. Remember, they are metabolizing the substance, and then when they release byproducts, then it makes it toxic. So if you are able to control the activity of the enzyme in the microorganism, then you can slow down the spoilage of the food. And so if you put food inside the fridge, or you put food inside the freezer, all you are doing is to put the food in a condition where you are slowing down the enzymatic activity in microorganisms that cause food spoilage. The same way with bacteria. Bacteria will be growing in food mediums that have much more water in it, say about 70, 80, 90% of water in food, liquid foods. Those ones, you have bacteria growing in them. That's a condition that is conducive for bacteria to grow, like milk. So if you put it inside the refrigerator, you slow down the enzymatic activity in the bacteria. So it isn't that the bacteria does not continue to grow in the food. That's the reason why you have expiry dates on foods, for instance. So that if you have the food even inside the fridge, it is not that the bacteria is not acting. It is still acting, except that it is acting at a slower rate. So that at a certain point, that food can become unwholesome, even though it is inside the refrigerator. And so you will also see that it is written on foods that when opened, and even when it is being refrigerated, you can keep that and consume it within a certain number of hours. Say 24 hours, you must finish using it. If not, it is possible that you have the growth of these microorganisms at a certain rate, at a certain level, which becomes, toxic to you. Right. So that is about temperature, and even with some application attached to it. Now, how about pH? pH, for instance. So here too, you see that the relationship of pH to the rate of the rate of an enzymatic reaction that an enzyme is catalyzing. The relationship looks very much like that of temperature. Now you can think of it this way that, for instance, in our mouth, we have salivary amylase that is acting, say, on carbohydrates, and our mouth is slightly, say, alkaline, or like slightly neutral. I mean, you have a pH of maybe around 6.8 thereabout to 7 point maybe 2 3 thereabout in the mouth, and that condition is optimal for, say, salivary amylase to be acting. But how about when you go to the stomach? In the stomach, you have hydrochloric acid, which is being secreted into the gastric juice, which means that the pH there is higher. There, you don't have the salivary amylase being active there. You have other enzymes that are acting, say, uh, pepsin, uh, enzymes within in the gastric juice that are acting on proteins. They are active at that pH, but they will not be active at a neutral pH like what you have in the mouth that salivary amylase is acting. So you remember, even from the second cycle, when you were studying metabolism or, say, digestion, they mentioned that in the mouth, carbohydrate is broken down, and then in the stomach, you find proteins being broken down. Then you ask, maybe it didn't occur to you, ask yourself, so how about proteins in the mouth, for instance? Maybe your teeth it is just that your teeth will be able to crush the food into pieces, but the proteins will not be digested because the enzymes that digest proteins are not in your mouth. They will not be active at the pH in the mouth. So pH is also important in the activity of enzymes. Right. Well, so for an enzyme that will be, say, active somewhere around neutral, obviously the relationship is such that if you increase uh, the pH to be more basic, it can cause denaturing of the enzyme. The same thing can happen also when you have the pH being reduced to go into the acid, to become an acidic medium. So, but this one, it depends on the type of enzyme, as I've explained. It is not like the temperature that you say it's a protein that is the enzyme. And so once temperature increases, that is going to affect all enzymes. It's going to cause them to get denatured. And if you reduce the, the temperature, it's going to inhibit the activity of the enzyme. In this case, it is enzyme specific. It depends on the type of enzyme you are talking about and where it is active. Some enzymes will be active in some basic, slightly basic mediums. Some will be active in acidic medium. Okay. Now we can talk about mechanisms of action of enzymes. Now, one of the models for studying uh, uh, the action of enzymes is referred to as the lock and key model. And in this model, it is assumed that the enzyme is having a certain shape such that it fixes perfectly with a substrate to act on it. And so you see it being depicted here that the substrate is being referred to as, is being depicted as a keyhole, or even let's call it a key. Okay, even if you say it's a padlock, it is still a keyhole in which you are going to put the key. So the key being the enzyme. So when the two bind, then it forms the enzyme substrate complex. And that enzyme substrate complex can then go into the the transition state so that a product is formed at the end of it. So you see here is being depicted as a key and a sub. This will fit more to say absolute specificity. But when you come down to the diagram below the substrate and the key, this is more like not absolute specificity, but you can say maybe group specificity or yes, group specificity. So it can be different molecules. So you see that there are some substrate molecules here. They have different shapes, and the enzyme is having different portions that even though these substrates are having uh, different structures, they are able to bind to the enzyme to give you an enzyme substrate complex, which can then go into transition state and the product is formed. And after that, so these two molecules actually now bind together with the enzyme acting on it to give you a product, and then the enzyme is dissociated from it. So you see this model uh, as that model which is uh, you know, talking of the fact that yes, a substrate and an enzyme should be compatible, fit so perfectly before the enzyme can be able to uh, uh, say catalyze a reaction that that substrate is involved in. But there's a problem with this model, and the problem is this. You remember that the last time when we were looking at the reaction profile of both non-catalytic and catalytic reactions, where we saw that an enzyme will cause the reduction of the activation energy which is involved, and that is how the enzyme will cause the increase in the rate of reaction of that's how the enzyme will cause increase in the rate of reaction. Now, I mentioned then that molecules will not undergo any reaction to form a product just on themselves because they just want to, unless they are unstable. And I made the example that if you are comfortable where you are, why will you want to change your position? Same way, if a molecule is stable at a certain state, why will you want to change into a different form? If it is stable there, it's stable. That is a comfortable state. That is a state of stability that it is in. So there will be no need for it to change, except there is some agitation, and that agitation is caused by the activation energy, right? So if that is the case, it means that if you have an enzyme being a key fitting to a substrate so perfectly, because a key fits very perfectly into a keyhole. And the moment it fits, it means it is stable. And if it is that stable there, then there will be no need for it to say to go into a reaction to have a product. So that was a problem with this model, the explanation of how the enzyme, acts in in chemical reactions. Now, that is depicted so clearly in this diagram, such that if this is a substrate which is being depicted like call it a stick or metal stick, and a transition state is the bent form of that metal stick, and then the product will be that the metal stick is broken, which means the enzyme is supposed to cause the breakage of this metal stick. So this is just a depiction to show, what I was explaining earlier. Now, if you have an enzyme, so the enzyme being this blue portion, So if you have an enzyme that is fitting so perfectly to that substrate, it fits so perfectly that there will be no way that you have the enzyme substrate complex getting converted into another product, because that state will be perfectly stable. It's just like a magnet that is getting stuck to it. And so it will not want to change into another form, which is a product. So then the understanding came that, okay, it means that at first you have the enzyme binding onto the substrate to give you an enzyme substrate complex, but that enzyme substrate complex is not perfectly stable. So it is that sort of that enzyme substrate complex itself is a transition state. So you see that this is not perfection. The fitting that you see with the enzyme and the substrate is not perfect. So rather, you see that it is after you have the substrate and the enzyme binding, then there is some kind of configuration, so that now you see that the substrate now kind of gets uh, configured to fit the enzyme. Then the enzyme can cause the breakage of that substrate to now give you the product. So then from here, with this understanding, then came the induced fit model. Induced fit model. Now, the induced fit model clearly depicts that a substrate and an enzyme are not perfectly fitting together. So you can see. So this time you saw that over there, when we were looking at this, remember when we were looking at this, it is the substrate that has gotten configured in the enzyme so that now the enzyme can act on it. But then here in the induced fit model, you realize that it is the enzyme. You see, it is being depicted here that, like, if this is the active site of the enzyme and it is round and the substrate is being square, it cannot fit into it. This square substrate cannot fit into this round enzyme. And so it is rather the enzyme that gets configured to now fit to the substrate. And then you have that enzyme substrate complex that now goes into the reaction locus. This is a transition state. Enzyme substrate complex goes into a transition state, and then further now reaction occurs, and then the products are released from the enzyme, and then you have the enzyme getting separated from the product. So you have this induced fit model, which is similar to what I was explaining earlier with the modification to the lock and key model here, except that here you see that it is the substrate which is being configured to fit the active site of the enzyme. Here it is rather the enzyme which is getting having some changes in conformation. So conformation of the enzyme is changed so that you now have the active site fitting to the substrate so that it can now catalyze the reaction on the substrate. That is the induced fit model. Further studies, currently, there is much more of understanding of the activity of enzymes, because there has been so many methods which have been used to study the activity of enzymes, yeah, especially studying the active center of enzymes to know which specific amino acids that are found, or that make up the active site of enzymes. There are mostly 2 or 3 amino acids that are close together in in an enzyme structure, which is mostly a protein that you have some 2 or 3 amino acids that are close to each other, and they're having some form of interaction between them such that when a substrate comes, they are able to interact with the substrate together to cause a reaction on it. In fact, now I mean, for a long time, there have even been computational methods for studying these things. Sometime in 2011, 2012, thereabouts, I was doing some research work on the active site of an enzyme, silicatane, which is an enzyme found in marine sponges, responsible for polymerization of some uh, silico compounds. You have this orthosilicic acid, I mean, silicon with 4 hydroxyl groups on it, and it can bring these molecules together to form like cubic forms of this molecule. And the active site of that enzyme, we could study it with computational models. So, and you could see, say, a proton transfer of, say, you have a proton transfer from one amino acid onto the other, which causes the enzyme to get active and begin to, react with uh, substrate molecules like an acid-base reaction form. Because remember that proton is a hydrogen ion which is being transferred. So it's just similar to an acid-base reaction which is occurring in the active site of enzymes. Now it has been known that uh, I mean, most amino acids will be having these specific most enzymes will be having these amino acids in their active centers. So in order of occurrence, you have histidine occurring more in the active center of enzymes, followed by cysteine, followed by aspartic acid, and then arginine and glutamate. So you find these, I mean, in more than 65% of the active site of enzymes. These are the amino acids which are found. And clearly you will see that, for instance, cysteine, I mean, cysteine is having a sulfur hydride group there. So it can easily, uh, you know, uh, undergo a proton transfer. The hydrogen which is present on the sulfur can easily be given out. Histidine is being shown here in an example, which is papain. So this is just to depict the structure of, so the green portion is showing like the full structure of a protein, which is papain. And then the active center is being depicted to show that, okay, this is cysteine and this is histidine. This number you see here is showing the, which is histidine 159, is telling you that the histidine residue, that amino acid, is found at the 159th position in the structure of the protein. So if you talk of the primary structure of the protein, where you are talking about the sequence of amino acids in the protein structure, on the 159th position, you have histidine. Similarly, cysteine, that amino acid, is found at the 25th position. So, which means the molecule actually starts from the top all the way down, because cysteine is at 25th position and then histidine is at 159th position. But the folding brings them close together such that now they form an active center, such that they are able to act on a peptide bond. So you see what is happening here. You see that you have the hydrogen of this amine group having some interaction with the nitrogen, which is in this ring of histidine. And you also see that the lone pair electrons on the sulfur, which is present in cysteine, is having some electrophilic attack, or call it that will be a neutrophilic attack, because these are electrons that will be acting, having a neutrophilic attack on the carboxylic part of the peptide bond, and in the end will cause the breakage. It's like stretching it. So you have this hydrogen bonding that's occurring over here, and you now have this neutrophilic attack, which is on the carboxylic group, causing sort of a stretch because of some stress on the bond, and then causing it to break. And that is an enzyme, papain. And papain is an enzyme which is used in the meat industry to soften meat, for instance. I mean, that's another application of the knowledge of enzymes in industry this time around, in industry. So you can see that all along we have been talking about applications of this knowledge of enzymes. So it's not only for the fact that that these enzymes are acting in biochemical reactions like digestion and metabolic pathways, but you can see that we have seen the application of these enzymes in various fields, like in pharmaceutical industry. I've just talked about the food industry, like the meat industry in softening of meat. And we also saw even in medicine, uh, medicine, pharmaceuticals, they go hand in hand. That is when we were looking at the muscle relaxant, succinylcholine. And we even talked about the knowledge being applied also in the storage of food. Right. So these are this is a really relevant knowledge that I think you should be appreciating. Right. Now, this is also to just depict again activity of enzymes, so that these are shown as some amino acids that are in the structure of an enzyme. And again, you see the numbers that are there. They are only showing that this amino acid is at 275th first position. This is at 295th, 209, 119. This is at 72, 269. So there will be that folding that it's not at because one amino acid is following the other. They are the only ones that are going to be close together. But you can have at different portions of the protein that, because of folding, you have amino acids at different positions that are coming so close together that they form an active site. So you see this active site is being depicted as some kind of a pocket, so that in that pocket you have a substrate coming in. So this place is being referred to as a hydrophobic pocket, where you have some CH3 groups, and you have some substrates that is coming in there. And you see metal ions as cofactors, magnesium ions, potassium ions. And this looks much more like a transfer of or the break of a phosphate group. So you see like two phosphate groups broken away from the first phosphate. Sort of you are having a triphosphate, that two phosphates are broken from it, and you now have a diphosphate. And the residue is a monophosphate. Or you see clearly this is an activity of a pyruvate kinase, which is being depicted. So pyruvate kinase, again, I mentioned that if you see the name of an enzyme with kinase in it, it means it is involved in the transfer of a phosphate group. And this is a reaction which is being depicted here, that this time around you have phosphoenol pyruvate coming together with ADP, adenosine diphosphate. So in the end you have pyruvate, and then you have ATP, which means the phosphate group, this phosphate group which is on the phosphoenol pyruvate, in addition to the two phospho the phosphate group, the phosphate groups in the diphosphate will now give you a triphosphate, and the pyruvate residue is left. It's just like this pyruvate is carrying the phosphate group, just uh, donating it onto the ADP. ADP accepts it and become ATP. And that is what is being depicted in this diagram here. Now, of course, this diagram is just I would say some computational image generated just to for you to see a pictorial view of how this thing can how when you talk of it, how if you look at it pictorially, you can have a better understanding than if you are just talking about it in a vacuum without any imaging. So this is just an image to depict how like metal ions like magnesium, potassium can be acting as um, some cofactors, and how you can have a transfer of one phosphate group, say phosphoenol pyruvate, onto a diphosphate adenosine diphosphate to synthesize ATP. Right. Now let's talk of another uh, application of the knowledge of enzymes. You realize that along the line I I I, I mentioned or some application of enzymes is because your course is structured in such a way that I mean you shouldn't study it in a vacuum. You should be able to apply it, uh, you know, into practical terms to really know that it is not just for the sake of the theory of it. So this is a, a medical application of the knowledge of enzymes, again in medicine. And this time around, it is on on the activity of an enzyme, protein kinase M zeta, which was found to be participating in a reaction that is relevant for long-term memory. So you have such protein which is involved in some reactions in the brain, responsible for the maintenance of long-term memory, things that you can keep in your mind for a very long time, for years. Then there has been the discovery of a drug molecule, which was called the zeta inhibitory peptide, ZIP. And what this drug does is it goes to inhibit the enzyme, the protein kinase M zeta, which is responsible for that long-term memory. So what is the importance of this? Now there are people who have very traumatic memories. People who have undergone, I mean, certain situations that are very traumatic, like, for instance, survivors of some very serious accidents or wars. People have been in wars, or even take for instance, like soldiers who go to war and I mean be at war for years, and they've seen a lot of people dying, and sometimes even their own friends dying beside them, their own colleagues and things like that. And all of these long-term memory causes them sort of trauma even after the war. Some have sleepless nights and so many other things concerning uh, mental disorders and things like that. But this drug is, if taken, can block the activity of such protein, the protein kinase M zeta, which is involved in long-term memory. So by that, the drug is able to manage the the memories of such people, so that there will be less less of times that they remember these things. That sometimes they're having sleepless nights, having sort of nightmares and things like that. So the drug is used to control that sort of a situation, that sort of a condition in these people. So you can see this drug again is actually developed based on the knowledge of an enzyme. So that is another application of the knowledge of enzymology. This time specifically on the protein kinase M zeta, which is involved in long-term memory. Now, so far, all of these that we have done, we've looked at, mechanisms by which enzymes act. So if you put all of it together, it means that in order to really understand the mechanism of action of an enzyme, you will need to understand things like the sequence in which the enzyme acts, say how the enzyme will bind to a substrate to form an intermediate. Then you also need to understand the structure of the intermediate which is formed itself at the transition state. And you can, in a reaction, you can have different transition states. So you can have multiple transition states. It's not that in a reaction you have just only one transition state, but you can have multiple transition states, which will mean that in order for you to understand the whole mechanism by which an enzyme acts, you must understand the structure of each and every transition state. And then how each transition state converts to the other. You have to understand those things. And also understand some thermodynamics, energy. So because we talk about the fact that there should be an activation energy, that tells you that every transition state is going to be at a certain energy level, which means there should be an understanding of the energy states of these transition states, the reactants themselves, and the product that is formed out of it. All of this will give you a full spectrum of how the enzyme acts, the full mechanism of it. But in fact, there hasn't been any enzyme that all of these things are understood. Some of them are, some of these will be understood, but not all. Even though now we have, like as I mentioned earlier, we have computational methods which you can use to study some of these things and see what can happen, especially for energy levels. So you can model the an enzymatic reaction. Say you have a structure of an enzyme. You don't necessarily have to take the full structure of a protein, large protein. No, you only need to know the amino acids that are present in the active site, and specifically know the type of reaction which is occurring. If it is some proton transfer, then you model that proton transfer onto a substrate, or the proton transfer of from one amino acid in the active site onto the other, which will cause the activation of the molecule. And then you have that reaction that will be occurring with the substrate. You can model these things and see the different energy levels that are in the system. But again, you will not be able to model each and every transition state. You may not be able to do that. And even the environment in which the enzyme is acting, say acidic medium, basic medium, to some extent, yes, you can control some of these conditions in a computational system to do this. But Of course, in a computational system, we refer to that as in silico. It will not be exactly the same as in a biological medium, say in vivo, to be different. And studying these things are difficult because the activity of an enzyme is very fast, so fast, such that fine, you may be able to slow down certain reactions with some inhibitors to be able to study the structure of uh, some intermediate along the line. If you let's say in a solution where you have an inhibitor that slows down a reaction, and you are taking the solution of the reaction, let's say every minute or every you may not be able to do that, say every maybe even 30 seconds. It's so short. Maybe you may be able to do maybe a minute or two, that you take the solution and then you put it into maybe some infrared spectrum or nuclear magnetic spectrum, and then you use that spectrum now to study the structure of that transition state. But of course, as you are doing even the measurement, still the reaction is ongoing, so you may not be able to get it fully. So really, it's a difficulty. You will not be able to get the full spectrum of it. You can get some, but not all. But of course, it gives some amount, some level, some adequate amount of knowledge of how the enzyme acts, which is useful at the moment. So I think at this point, the information which we have gotten now is a lot. So I think we don't need to add more to it. The next time when we meet, we will look specifically at some mechanisms of reaction, like the mechanism of action of chymotrypsin. We'll look at mechanism of action of lysozyme in breaking bacterial walls, still mechanism of lysozyme along the lines. So we'll look at that. But for now, we'll end here. Thank you for your audience, and I think we can now open for questions and contributions. So if there are any questions and contributions, you can put up your hand, and then I will allow you to unmute yourself. Then you speak. So the floor is open. The floor is open, so you can put up your hand if you want to ask a question or you want to make any contribution. You can put up your hand. It's 1:35. Let's do it quick. Maybe in the next 10 minutes, if we can take questions and contributions, then we close. All right. So if nobody has anything, then I think I will. Okay, Abigail. Abigail, you can speak. You can unmute yourself and speak.
Sir, please, I just wanted to ask about the slides.
Slide.
Yes.
As always, you will get them.
Okay.
You will get a video too.
All right. All right. Thank you.
You're welcome. Yes, anybody else? Abigail, you can put your hand down. Okay, I see Courage. Where's Courage? Courage, you can unmute yourself and speak.
I wanted to ask.
Mhm.
You said there was an enzyme that can help people like forget stuff, like works on
Is there what? Is there? I can't hear you. Is
you
there what?
were saying there was an enzyme that can help people forget stuff, like forget long-term memory. So does it mean that our body like naturally produce that enzyme also?
Okay, so um, it is not an enzyme that, if I get you well, you are saying, is it there an enzyme that can make people forget things? The enzyme that we talked about, well, it's not known whether there is an enzyme inside the body that can cause us to say not to remember things. If there will be, say, maybe a protein in the system, say an enzyme that is responsible to cause us, like say internally, then it means that activation of that protein in the body can cause, the loss of long-term memory. It means that one, it will be internal. You don't have to take in any external drug. What we talked about was an enzyme which is responsible for the long-term memory, and then a drug which is a molecule discovered to cause inhibition of that enzyme can be taken in, so that it goes to block the activity of that enzyme, which then results in the loss of long-term memory. But it will be interesting that maybe in future, if it is found that there is actually say an internal protein, say an internal enzyme in the body itself that can cause such a thing, it means that there will not be the need to even synthesize some artificial substance which has to be taken in to cause that, but you can just cause the activity of that. But all the same, you will need to take in something external to go and cause that activity inside there. I hope it's clear.
Forget stuff. There was something that was important.
Courage, I can't hear you well. Courage, were you saying something? Okay, I think it's just some noise coming from him. Yes, any other question, contribution? Yes, Philip. Philip, you can unmute yourself and speak. Philip, you can unmute yourself and speak. Or Philip didn't want to ask a question. It's by mistake that his hand is up. Okay, Philip.
Let's say, yes, sir. Please, is there a class of enzymes known as the translocases?
Known as what?
Translocases.
Well, that will
Within the
just be
same chapter.
that will be the same as transferases.
Okay.
Mhm. Yes, any other question or contribution? I love to have contributions so that you can share with us some experience, some of you who have been working at some places and you know about the activity of some drugs which are based on enzyme activity, or even in the food industry, some practice you have which is related to enzyme activity. You can share with us. All right. It appears nobody wants to talk and you just want to go and enjoy your Sunday. So I will propose that we bring the class to a close. So if you are in agreement, you can put up your hand to show your secondment of the motion. Then we can close for today. Till the next time. Okay, Emmanuel Mensah is the first person. Courage also seconds. And then more of you. Is it Bernice, McCaffrey, Joseph, or Josephine? Alfred, Ferdinand, Daniel, Ernestina, Bright. Okay. All right, so then all the best and see you the next time. Bye-bye for now.